All right, I am going to talk about DNA structure and replication. So DNA is also known as deoxyribonucleic acid. If you can learn to say that, you can probably impress some people. But DNA is, like I said, what we use very short for the actual scientific name, which is deoxyribonucleic acid. And it is the genetic information that carries information about an organism from one generation to the next generation. So when your parents had you, they passed along their DNA to you, and when you have children, you will pass along your DNA to them. DNA is found in chromatin in the nucleus during most of the cell's life. So whenever it is not getting ready to replicate or not getting ready to go through mitosis, the DNA is found as chromatin. During mitosis, the DNA is condensed into chromosomes that we can see. So remember, these are the chromosomes, and chromatin looks like a bunch of stringy stuff. DNA molecule looks like a twisted ladder, and we're going to build one in a sec so you'll be able to look at it. But it looks like a twisted ladder, and it is made up of units called nucleotides. Stop and say that really quick. Nucleotides. Okay? Each nucleotide is made up of three things. It has a 5-carbon sugar, meaning it looks like a pentagon. One has 5 sides. Okay? It has a phosphate group, which is like a little circular thing, and then it has a nitrogen base. Now, there are a couple different types of nitrogen bases, and we'll look at those in a second. But this is the basic building blocks of a DNA molecule. Here's a DNA molecule. You'll see we have the 5-carbon sugar, the base, and the phosphate. And look, they hook together. So this 5-carbon sugar hooks to another phosphate, which hooks to another sugar, another phosphate, another sugar, another phosphate, another sugar. And the sugars and phosphates are what are known as the backbone, okay, or the sides of the DNA ladder. All of the bases, look, this base hooks onto this base hooks onto this base to this base, this base to this base. These are the rungs of the ladder. So the nitrogen bases make up the rungs, or the middle of that DNA ladder. Nitrogen bases. So DNA has 4 kinds of nitrogen bases. We have adenine, thymine, guanine, and cytosine. When they're pairing up like this, okay, so when we have one string of nucleotides and it pairs with another string of nucleotides, okay, the rule is that adenine will only pair with thymine and cytosine will only pair with guanine. Okay, so A only goes to T and G only goes to C. During DNA replication, so remember, during part of interphase, we copy our DNA, and that's what DNA replication is. Okay, DNA replication is when we copy our DNA. Basically what happens is the existing DNA molecule gets unzipped. So it's as if we take a zipper and we go zzz, and unzip it into 2 separate strands of nucleotides. Okay, what happens then is there is a new strand that forms because new nucleotides come in and pair with those other nucleotides that were on the DNA strands. And the result of that are 2 identical DNA molecules, okay, 2 identical DNA molecules. So you'll see the red one is the new strand, the blue one is the old strand, okay, and they will become absolutely identical, okay. So I'm going to show you really quick what this might look like. I think it's super important that you see an example of it. So this is a gizmo called building DNA. So you'll see we have the building blocks of our nucleotide. So I'm just going to bring out, this is our 5-carbon sugar, our 5-sided sugar. Here's our base, but what we're missing is our phosphate. Okay, what we're missing is our phosphate. So phosphate, 5-carbon sugar, and a base, that is a nucleotide. Okay, that is a nucleotide. All right, so to make a long string of nucleotides, all we have to do is, it doesn't matter which base is attached, we'll just keep attaching new 5-carbon sugars and phosphates and alternate them, like that. Okay, it doesn't matter the order for the time being while we're just playing with this molecule. Okay, doesn't matter at all, but what you are going to do, is eventually we are going to have this whole DNA molecule all nice and built. And notice that it only has one side, right? So if this were a ladder, it'd be a pretty crappy ladder because there is no other side. It's just one side of the ladder and then a bunch of step stairs, which if you tried to put that down would not go very well, right? The ladder would just fall over. So DNA, one of the perks of it is that it is, is that it is double-sided. So now you'll notice that we have nucleotides that face the other way, and we have to pair them so that we have our complete ladder. So if you remember what I just said, okay, about the pairing rules, you should remember that A is going to pair with T, or adenine is going to pair with thymine. So when I put them together, they stick. Then we need a phosphate, and then if you remember, G pairs with C, and when we put them together, they stick. Now I want to show you, if I try to pair, let's say, G with A, notice that it wouldn't fit, right? It wouldn't go together like a puzzle. Each of those two molecules are too big, okay? And if I try to pair C with A, notice that the shape of the molecule doesn't fit together like a good puzzle. It won't fit. So that's why A has to pair with T, because it's small enough and has the right shape. And G has to pair with C. Hold on, let me add a phosphate really quick. Okay, G has to pair with C so that the molecules are the correct puzzle pieces for each other. So if you keep doing this, remember, you're alternating a phosphate and then a nucleotide, a phosphate and then a nucleotide, and you're doing it and making sure you're pairing up the correct nucleotides with what is across from them, and this is how you end up making a copy of DNA. Okay, so like that and like that. All right, so now if we were to watch it actually replicate, we're going to take this enzyme and it's going to unzip our DNA. Ready? So it unzips it, pulls it apart just like that. Okay, now what's going to happen is we are going to be able to pair. So if you look at this side and then look at this side, they were perfect matches for each other. So when we actually bring in new nucleotides to make the new strands on each, they're actually going to be the same. So we're going to look here at this C right here, and that will pair with G. So we're going to bring it in right there. Then we are going to bring in an A and another A just to finish, making our puzzle. Okay, making our puzzle. We are going to bring these in, T, C, T. Now really quickly, I want you to look. I want you to not look at this really quick, but compare this side to the original DNA molecule, this side of the original DNA molecule. So G, G, A, C, C, G, G, T, T, C, C, T, T. Look, they're the exact same. So this new side is identical to what was there before. Okay, the new side is identical to what was there before. So when we finish making this other side, okay, so C, T, T goes with A, G goes with C, C goes with G, A goes with T, G goes with C, and A goes with T. Look, we are going to have two completely identical, if you look at them, they are identical molecules, okay? And that's good. That means now we have two complete copies of our DNA. So that's kind of what happens when DNA is replicated, and that's kind of the structure of DNA. So I hope that helped make more sense of it. If you want to play with this, just log on to your Explore Learning username and password, and the gizmo is called Building DNA, and you can play with it tonight. It's super easy and fun to play with. Feel free to do that. It is fun. We will be doing a couple of, not the actual gizmo, but a different activity tomorrow. But this is a really good practice for you to kind of start to learn and understand the structure and function and replication of DNA. So thank you. I hope that made sense. Write down any questions you have.